session. So we're going to round out the clinical trials session and really focus on the patient perspective. So first, Dr. Riccardo Perfetti is going to give a short talk on an update on the phase 3 INSPIRE clinical trial for SORD deficiency. And then I'm going to ask him a few questions, and we're going to open up to the audience for questions about clinical trials from the patient perspective. So I'm first going to introduce Dr. Perfetti. So Dr. Perfetti received his MD and PhD degrees in endocrinology from the University of La Sapienza in Rome, Italy, and completed his postgraduate training at the National Institutes of Health in endocrinology and molecular biology. He then practiced as an endocrinologist at Cedars-Sinai Medical Center and performed research as an associate professor of medicine at the University of California, Los Angeles, and as a senior staff scientist at the National Institutes of Health. Dr. Perfetti transitioned to industry in 2004 and worked as a director and global development leader in diabetes, obesity, metabolism, and endocrinology at Amgen. In 2007, Dr. Perfetti joined Sanofi and served as senior medical officer, vice president, and head of global medical affairs in the diabetes and cardiovascular business unit. Dr. Perfetti joined Applied Therapeutics in his current role as chief medical officer in 2018, and he's here to share an update on their clinical trial for SORD deficiency.
Thank you, Kathy. I heard in several sessions a SORD study to be mentioned. We are very fortunate to be already working on the pathway that in patients with SORD deficiency is affected. And we are working on this pathway for different diseases. And we immediately realized the opportunity to start investigating the potential benefit for SORD. So patients with SORD have a mutation on a gene that is called sorbitol dehydrogenase. The disease is an autosomal recessive disease. Most patients develop the clinical symptoms in their teenager years. Although new paper seems to now move the age of onset a little earlier than that. The gene defective is on a gene that is on a very short, a very secondary pathway to utilize glucose. Glucose is a major substrate for energy production in many species, including humans. And the most we do out of glucose is to transform it. And in the process of transforming, we produce energy. Another way to utilize glucose is to store it and eventually use it to produce energy. Evolutionary, we have another pathway for which there is very little use, is called the polyol pathway. About 3% of glucose in humans is shifted through this pathway. It's a very short pathway. There are only 2 steps. The second step is the one that is modulated by this gene, which is called SORD. The SORD transforms 2 sugar product, one into the other. It transforms sorbitol into fructose. Because of this gene defect, patients with SORD deficiency accumulate this byproduct of glucose in levels that are 100-fold higher than in normal individual. We are investigating means to block the making of sorbitol so that in the setting of a reduced sorbitol dehydrogenase, you have less substrate to work with, and therefore you will be able to reduce the toxic effect of sorbitol. This is the pathway. This is a little bit of biological chemistry. Fortunately, it's a very easy biological chemistry because it's a short pathway. There are only 2 steps. In animal model where you can delete this pathway, so abolish the pathway, the animal actually live longer. Now, animals are not identical to humans, but that confirms once again that this pathway for us as human may have a very neglectable, if any, role. So the second step, which you see labeled as SORD, is the one that in patients with this gene deficiency is affected. Our intervention is one step earlier. We try to reduce with a molecule that is called 80007 or govorestat, the making of sorbitol. So as I mentioned when I started, when the publication from the group of Dr. Zuckner, first author Andrea Cortese, was published in 2020, came about, we were already working on this molecule. So much of the work done in terms of in vitro characterization, animal toxicology, phase 1 study in healthy volunteers, dose ranging was already done. So we were relatively lucky, we perhaps as community of physicians, scientists, and patients, to be able to immediately initiate the clinical study in patients with SORD. So the very first step actually was to see whether cells obtained from patients with SORD deficiency indeed in vitro had a very elevated level of sorbitol. The graph that you see on the left of the figure with the big bar is the level of sorbitol that you see in patients with SORD deficiency. You also see the level in healthy volunteers. You see the degree of reduction that we induce when we, in culture, in a Petri dish, we add the molecule that inhibits the pathway. So in principle, the molecule should work in patients with diabetes. What you see on the right side of the slide is the animal study. I don't know if I have a pointer. Is this also a point? This is probably a pointer. Okay, very good. So the animal study, it's an unusual animal study. This is a Drosophila study, so it's the so-called fruit fly. The fruit fly use this polyol pathway also, but not with a biological need. So we were able to engineer the gene in the fruit fly. And what you see, this is the neuronal structure in a control Drosophila, and you see how disrupted is in the animal that has the SORD deficiency. Treatment of the Drosophila with this inhibitor of the aldose reductase is able to preserve the neurological structure. So this is not a rescue experiment. The animal were treated from the early days with the inhibitor of aldose reductase, and we don't see that disruptive path. With the Drosophila, the animal doesn't only fly, also walk. You can measure how fast the animal walks and the direction that takes. So in animals that are normal, this is the speed. The animal that is deficient for SORD runs at the very deficient speed. When you actually treat them with this drug, you can almost restore the actual speed. This is the direction. The animal that is deficient for SORD does not find the way to climb directly. They're placed in a jar, at the bottom of a jar. They climb on the wall of the jar, and they climb straight up. The animal that is deficient for this gene trying difficult to find a way out. They keep falling on the bottom of the jar. But the animal that has been exposed to the study drug seems to find the way out. Now, this finding the way out occurs obviously in a setting where sorbitol is reduced, but it's not reduced to the level of control Drosophila. So there is a very large reduction, but there is not a real total normalization. There is a 60-70% normalization. Now, this is the first study we did in patients. There were 8 patients participated to this open label pilot study. The objective of this study was to see whether the drug was safe in patients with SORD. This study took place once about 200 healthy volunteers were already tested with the drug, and the drug was also tested for another disease, which is called galactosemia. But the objective of this study was, first, is the drug safe? Does the drug work? And what you see in gray is the level of sorbitol of the patient when they enter the study, and in green, the level of sorbitol at the end of the study. Actually, this green bar was the result after only 7 days, but from 7 days to 90 days, there wasn't much of an additional change. So the drug worked and was safe, and this was the basis for the dialogue with the FDA, the opening of the IND, and the initiation of the phase 2/3 study, which is what you see in this graph. So we immediately asked the FDA whether we could have compressed the clinical development approach and develop a study that will assess multiple different things. Patient enrolled in the phase 2/3 study needed to have the genetic diagnosis of SORD. They needed to have an elevated level of sorbitol. Their age was between 16 and 55. In the United States, 18 to 55, because the FDA objected to patients of age 16, although that's really when the disease presents itself. These patients were stratified based on their severity of the disease. That was evaluated on the performance on the 10-meter walk and run, which is one of the components of that very large test that you heard about today several times, called CMT-FOM. So there were 3 categories: mild, moderate, and severe. We stratify active and placebo. So this is a placebo control, 2 active and 1 placebo. In the very first part of the study, we primarily monitor biomarkers, of course, also safety. At month 12, we do the first outcome assessment, which is the full CMT-FOM, which includes the 10-meter walk and run, the muscle MRI. You heard several times about the muscle MRI, the pool of biomarker. The study continues and reads on month 24, although the month 12 is an interim analysis. If the study is successful on month 24, the patient on placebo will be offered the opportunity to be transferred to the active drug. The one on active drug will probably offer to be part of an expanded access program. At baseline, there are some very interesting observations. So at baseline, the level of sorbitol seemed to be correlating with the severity of the disease. The highest the sorbitol, more severe is the level of the disease, which was measured through 3 different measures of clinical evaluation of patient. In a cohort fashion, the 3-month analysis revealed that the active group show a very significant lowering of sorbitol at 3 months. This investigation was conducted by the DMC at month 3, when patients have been exposed for a sufficient amount of time. You see that the patient exposed to the active drug have a very significant lowering of sorbitol. This is the level of lowering that you see in placebo. So patients with SORD deficiency have levels that are between 25,000 to 40,000 nanogram milliliter. Healthy individuals that don't have SORD have level around 200 to 300 nanogram milliliter. So it's a very gigantic difference to the point that perhaps SORD could become a critical component of the diagnosis of the disease, in addition, obviously, to the genetic, or sometime in place of the genetic, because the genetic for SORD is a little bit complex due to the fact that there is a pseudogene that sometimes interferes with the identification of the gene defect. So we have seen that during the study, during these 3 months, we were able to move large part of the patient from this level to levels that are significantly lower, and in some cases are levels that are similar to the heterozygous relative of these patients. They're not as low as in healthy volunteers, but they're very similar to the one of their parents or their siblings that are heterozygous. Most of the sites that participate to the phase 3 study are part of the INC. We have a total of 10 sites, including 3 in Europe, mostly in the United States. And I think this is my last slide. And as you can see, things are moving fast. And hopefully we're going to be able to communicate more substantial data very soon. Thank you for your attention.
Thank you, Dr. Perfetti. So I have some questions here, but I know we're short on time, and I want to make sure the audience has time to ask questions. So maybe we just jump to Q&A of the audience. Questions? All right, well, how about I ask one to start? Okay, will patients need to have a genetic diagnosis to participate in a clinical trial? And we talked about this a little bit earlier, but your thoughts on insurance covering genetic testing?
In this study, we provided all patients with genetic testing, even if they came with history of genetic. And we also performed the sorbitol measurement. Yeah.
So it seems like maybe those initial evaluations will be included in the actual trial process.
Correct.
That's
yes.
Great. Yeah. Questions? Yep.
But I got a question. How many, or it's twofold, how many SORD patients are there in the world that you're attacking? So I might have missed that, but if you can answer that. Do you have an answer? And then I'll follow it up.
How many patients with SORD deficiency? So in the United States, the estimate is that is around 4,000 patients with SORD deficiency.
Great answer. So everyone in the room that doesn't have 1A, take a look at what's going to be solved. One of the rarest types of CMT. Thank you.
Looks like I don't have it.
If I'm a patient who doesn't receive my care at a CMT center of excellence, but I learn about a trial, or I want to learn about if there is a trial, what's the best way to do that? And can I participate even if my primary neurology care is not typically at one of those centers?
Do you mind rephrasing?
Yeah, so she's asking if her primary doctors that she sees for her neuropathy aren't part of the centers that are actually performing the clinical trial. How does she learn about clinical trials? And
yeah.
Take part?
Sorry, for the acoustic wasn't too ideal, or maybe my hearing is not right. So as often in rare diseases, the network between patients and physicians is fundamental. And I think the spirit of event like this, for which I really have to commend the organizers, are to really continue foster the relationship of the patients that are part of this study. I would say only 1/3 came through direct referral by their own physician. Most of them were referrals to referral and eventually referral to me, as oftentimes patients with rare diseases, there is that very savvy attempt to gain any potential information for novel therapies. So you are here to obviously testify the fact that you are part of management of the disease. So I would say a large part of the patients that are in this clinical study asked their physician, and their physician was humble enough to ask other physicians about what is new. Many identify the study by surfing the web because it's posted on this repository that is called clinicaltrial.gov.
All right, maybe one more question before we move to, oh, we got a couple here. Maybe let's do these two and then we'll move to break.
With these two?
Yeah.
Thank you. Thanks for the presentation. I have a question. So my name is Allison Moore. I'm the founder of the Hereditary Neuropathy Foundation. We worked with the Zuckner Lab to make the SORD deficiency rat, and he's characterized it. He's in the process of publishing. I'm curious if this rat would be useful for more data from a regulatory standpoint, or as you're moving forward, hopefully, to a successful drug.
I missed the first part. Say the first part again. Do they need more preclinical data to
I was curious
the
if you need
regulatory component?
Yes,
yeah,
yes.
That's the question. So do you think the regulators will be convinced with just your data alone, or do you think you need more preclinical work with SORD deficiency?
Well, we need to work on this all together. I've been working on developing drugs for at least half of my professional career. First was academic, the second in the industry. The FDA has been structured and educated, built on the more stereotypical type of development, on the development based on evidence-based on long-term clinical evidence trial. And I was an investigator of this type of trial. Obviously, when a disease affects few people, in principle, you should never start the study because you're never going to be able to demonstrate, you're never going to have enough power. So I'm not going to go into a long discussion on this, although I would love maybe between the break to have that conversation. To answer more directly your question, I believe that the point we'll continue to make into the agency is that the complexity and the entirety of data may need to be reviewed, not necessarily the long-term clinical outcome. This is a slow progressive disease, as many other forms of the CMT. This study cannot last for 20 years to show the endpoint clinical outcome. So we will continue having an open dialogue with the agency, and the agency is receptive to the fact that there are diseases that are different from diabetes and hypertension, for which you can have a study that enrolls 1,000 patients, and in 3 months, you're able to tell whether you're lowering blood glucose in a patient with diabetes. But in a disease that evolves over decades, you really need to be creative and be more science-based. That's why this study was equipped with biomarker, MRI, patient-reported outcome, patient satisfaction. So it's a very rich study. So I am optimistic that the FDA will continue to be receptive to the evidence, but also to the advocacy that may come from people like you.
Last one here.
One second. Sorry.
You mentioned one precondition was age between 18 and 50. Are there other conditions that would exclude you from testing that you have to look for?
Other physician?
Yeah, he's talking about inclusion and exclusion criteria for the study.
No, the criteria of inclusion and exclusion were primarily aiming at identify patients that were not too severe and with yet symptoms. But this is not because I believe that the drug may not be beneficial to all, but rather because of the constraints of showing change. So the only criteria that were dominant in the study for inclusion was the diagnosis. For exclusion was the presence of comorbidity that were so severe they would put in danger the life of patients. So to have another condition was not per se an exclusion, only if that condition was such that the well-being of patient was at risk during the study. So the study was very inclusive, but not inclusive to all, because we were obliged by the regulators to show short-term benefit.